right, this is mitosis notes part 1. And materials you're going to need will be some paper. Blank paper is best if you have like printer paper at your house. Something to write with, something to draw with, colored pencils. If you don't have those now, you can always go find them later and add to your notes. Just don't forget. Title your paper Mitosis Notes Part 1. And then we're going to be drawing some circles on the left side of the page to represent animal cells. Remember that animal cells are round-ish in shape because they lack the cell wall that a plant cell has. I'm going to use this glass as my template for my animal cells. You can freehand it if you want, but yay, go Lions baseball. And just make sure that since we're going to draw 3 on this page, I want to make sure that I can fit 3 right along here. So I'm going to draw the animal cells over here. I'm going to write notes about each stage over here. So I'm going to start pretty high on my sheet. It doesn't have to be perfect. Once again, you can also just freehand it if you don't have a circle template handy. It's not a big deal. Okay, there's one. I'll just fill in a little bit. There we go. Okay, here's my second one. And my third one at the bottom. Okay, there we go. So if you need more time, pause the video and then turn it back on when you get caught up. All right, looks good. Okay, so we're going to start with interphase. I'm going to label it below because that's where I have the most space. So this is going to be a drawing of a cell in interphase. Remember that term that was part of our cell cycle that we sketched. This one is going to be labeled prophase. Sorry about that, prophase. And we've had that prefix before. Pro means before or early. Prophase. And then our last one on this sheet is going to be labeled metaphase. Meta, like as in middle. It's the middle phase, and also there's something happening in the middle of the cell. So that's a good name for this phase. It kind of makes it easier to remember the name of this phase. Okay, so here's what we're going to do. I'm going to do the sketching and then the notes for each picture. And so follow along with me again. Feel free to pause as you need to. I'm going to choose a color to dedicate to each cell structure that I'm drawing in the notes. So I'm first going to choose a color for my nucleus, and I'm picking this kind of indigo purpley blue. And so here it is. This is the nuclear membrane. Remember, the nuclear membrane is important because it protects the DNA, the genetic material of the cell, from all this crazy stuff happening out in the kind of the wild west of the cytoplasm. All right, so let's label these as we go as well. Nuclear membrane. All right. And so, like I mentioned, inside the nucleus is the DNA, the genetic material that has the codes for everything that the cell does. And so I'm going to color it. I'm going to pick this light blue color. And so everything I do from here on out that has to do with the DNA and the genetic material is going to be in this color. So I'm going to just kind of represent the DNA inside with these sort of loose kind of like yarn, sort of unraveled, kind of like squiggles, not very organized. In this form, as we reviewed already, the DNA is called chromatin. So it would be kind of like if you took a ball of yarn and you unwound it, and it's just in like a big heap, and it fills up a larger space, but it's not very organized. So there are proteins, there's DNA in here, it's just not very compact. It's just kind of filling up the space it's protected within that nuclear membrane. So that is chromatin. And then we're going to draw a dark spot over here to represent the other organelle that we just kind of talked about, which is the nucleolus, the site of ribosome assembly. So most of the time, if you have stain on a living cell or a dead cell that's been prepared, you can often see, if you have a good microscope, you can see a dark spot inside the nucleus that represents that nucleolus. So we're going to sketch it there. All right, and then two more structures we're going to add here. I like to use brown for these little guys, the centrioles. And they look like little logs, so I like to make them brown, like little fire logs. And as we just reviewed, the centrioles are organelles that are going to assist with this whole process of cell division. All right, so interphase. I'm just going to give that some color to make that pop. All right, and of course, the outer boundaries here of our circle, this represents the cell membrane. All right. So when you think about this word, inter kind of means in between. So it's kind of the in-between phase. All right, so let's jot some notes down over here about interphase. So first fact is it's the longest part of the cell cycle. Okay. And we mentioned in the cell cycle notes, I mentioned to you that there are some cells that go through interphase and then they just don't divide. So they're what we call non-dividing cells. Do you remember what those were? Cells that don't grow back if you damage them. So that would include things like muscle cells, nerve cells. So non-dividing cells exit at G1 of interphase, and that was nerve and muscle cells. Okay, and so remember on our sketch notes, let me grab the right one. On our sketch notes, we had that interphase was made up of G1, S, and G2. So let's put those in our notes as well. Make sure I have this straight for you. Okay, so G1, remember, stands for gap 1, is when the cell's growing, so G for growing, and they just do their job. So if I'm a skin cell, I'm protecting from foreign invaders. The next step of interphase would be S, and remember S stood for synthesis. And synthesis means to make or to create. So this is when the cell copies the DNA. Because remember, the whole goal of mitosis is to start with one cell and end up with two identical daughter cells, so cells dividing. And in order to do that accurately, everything needs to be a perfect copy within the two new daughter cells. So the DNA needs to be copied, and that takes place during interphase, the S portion. Okay? And each chromosome, we have some vocabulary here that we just reviewed. Each chromosome gets a sister chromatid. So that loose chromatin starts to condense into these stick-like structures called chromosomes, and it's duplicated and becomes sister chromatids. So two exact replicate copies stuck together, sisters. Okay, G2 was gap 2. And this is when the cell's resting and does its job. It's just getting ready for that big division because that's a lot of work to divide. All right, very good. Let's keep going. So that was interphase, lots going on, longest phase. And let's move on to prophase. So I'm going to switch back to my color pencil mode. I'm going to grab my kind of indigo color I used here for the nuclear membrane. And I'm going to draw it a little bit differently this time. I'm going to draw it with a dashed line because during prophase we said means early or beginning. During prophase, the nuclear membrane starts to break down. So we draw it like it has these little holes. It's disintegrating. Okay, some other things that are happening. We mentioned that the chromatin was condensing, and at the end, everything was getting copied, and we have these sister chromatids. And now in prophase, they have condensed so much that we can actually see these pairs of exact replicate chromosomes called sister chromatids visible inside the nucleus. And so we have these sister chromatids, and they are just kind of separated out. They're not all relaxed, they're condensed, they're tightly coiled along with their proteins. And then something's happened with our centrioles. Our centrioles have started to move to the opposite sides of the cell. And the opposite sides of the cell have kind of a fun name. They're called the opposite poles, kind of like we would think about a globe. We have the poles, the North Pole and the South Pole. So we have the poles of the cell, so the centrioles have started to move. And we talked a little bit about how the cytoskeleton is kind of this sort of invisible structure made up of these long strand-like proteins that help protect the cell. They help it, give it some structure, they help with movement. Well, those long strands of proteins make up most of the structure of the cell, and they actually are what is going to be coming from these centrioles. So these long protein tubes start to form, and they have a special name. Think of the centrioles being kind of like a rod and reel, and it's kind of helping to organize these fibers that are kind of shooting out of it like a rod and reel. And so we're going to label these just to make sure we remember these are the centrioles. And then these protein strands are called the spindle fibers. Spindle fibers. So here's our nuclear membrane. Here are our condensed sister chromatids. And I think that's all we really need to label on that one. Let's put before or early. So it's before division has actually happened. It's kind of that first step in the nucleus dividing. So let's highlight this one also. We should go ahead. Let's label this at the top as well right here. Interphase. Just to make sure it's clear. And I'll do the same thing right here. So we're going to label this one prophase. Put a little box around it. So all these notes are going to be about prophase. Okay, prophase, the chromosomes are condensed. And become visible as sister chromatids. The nuclear membrane breaks down. The spindle fibers form. The centrioles move to opposite ends of the cell. And remember, we call those the poles. All right, and that is prophase. Okay, make sure you can see this. I'm actually going to bump it up just a little bit. Okay, so metaphase. Ooh, this is when it gets fun. So remember I said meta, I think of middle, because it's kind of the middle phase, and also something exciting is happening in the middle of the cell. All right, so let's start by drawing our centrioles. They're kind of anchored in the same spot. Remember, they were shooting out these long, flexible protein fibers we called spindle fibers, and they're going to play a huge role in the next step. So our nuclear membrane has completely disappeared. It's broken down. And our sister chromatids have done something kind of interesting. Remember to use the same color for your sister chromatids as you did the DNA material before. They have lined up in the center of the cell. If we call the ends of the cell the poles, can you guess what we're going to call the center of the cell? We're going to call it the equator. Okay, I'm going to go back and add some little dots to the center of these sister chromatids. We didn't talk about them yet, but I'm going to talk about them now. These are called centromeres, which just means center location. And you can kind of think if you had two pipe cleaners and you wanted to hook them together, you could put a paper clip in between to kind of hold them together. And that's kind of what the centromere does. It's holding these sister chromatids together. Let's label them just one more time. And let's label our centrioles again. Okay, and so something really cool has happened here. Our centrioles had started to shoot out these spindle fibers, and some of them are just kind of anchoring fibers, and then some of them shoot all the way out. Remember I said think of it a little bit like a rod and reel. It's going to shoot these long fibers out. Think about casting a rod, and they're going to go all the way here and attach to the centromere of the sister chromatids. So shoop, 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 just like that. And same thing over here. Isn't that cool looking? All right, there we go. Metaphase. So who's lined up in the middle? It's the sister chromatids. All right, let's write some notes about metaphase. All right, so the sister chromatids line up in the center of the cell. And remember, our word we can use for that is the equator of the cell, which is kind of fun. Next, the spindle fibers that are associated with the centrioles attach to each centromere. And this is the shortest part of mitosis. So if we were to take that cell cycle pie wedge and break it down between interphase, prophase, metaphase, and the other phases that come after it, metaphase is a tiny little wedge, tiny little sliver. Doesn't take up much time. All right, so before we stop for today, I just wanted to review some of this terminology because I know there's a lot of words that sound alike and a lot of new terminology. So I just wanted to make a little key down here for you. If you have space, or you can put it somewhere else if you need to. You can even flip over your page if you need to. So chromatin, kind of make a little key for ourselves. Chromatin is the relaxed DNA. Just get this up just a little bit. proteins. We'll talk a lot more about DNA and DNA structure coming up in our next unit. Okay, and when it condenses, these proteins help kind of act like little bobbins for it to like spin around and twist around and kind of hold it together in a tightly condensed format. The DNA forms into what we call chromosome. So this was chromatin, this is chromosome, which is condensed DNA. And it kind of looks like a stick. Okay? And again, we'll learn a lot more about chromosomes in our DNA unit and our genetics unit. And then sometimes we see the genetic material portrayed like this. And so remember, this is what we call sister chromatids. And this is just a duplicated chromosome. So it's like this one just made a mirror image of itself, and they're being held together by this centromere. So it's like they're paper clipped together. So each one is a mirror image of the other, and that's intentional because when the cell divides, then it can split apart. Half can go to each new daughter cell. So chromatin, chromosomes, chromatids, same stuff, just different formats. And that's where we're going to stop for today, and we'll finish up Mitosis Notes Part 2 next time.